My talk is entitled SARS-CoV-2 Vaccines, A Brief History of Vaccine Technology.
Much of this presentation is derived from a paper we published in early 2021. This diagram is from the publication. It shows the 4 different vaccine technologies that have been used to make the COVID vaccines. 2 of them are old technologies and 2 of them are new. Here is a link to the publication, which you can read at your leisure. To explain the different technologies, I've chosen to show you the historical background of who invented these ideas and when.
So, for example, we'll start with Louis Pasteur and the inactivated virus vaccine, and then we'll move on to Alexander Glennie and the subunit vaccine. And then we'll talk about Oswald Avery and the nucleic acid-based vaccines, and that includes Frank Graham and the non-replicating adenovirus vaccine, and Giorgio Dimitriadis and the messenger RNA vaccine. So let's start with Louis Pasteur. Everyone knows about Louis Pasteur. He has many inventions. He invented, of course, pasteurization. He saved the silkworm industry in France. He had some chemistry inventions. But what we really remember him for was his work in microbiology. And he was one of the leaders in identifying this new germ theory that many diseases were caused by these microscopic things that you could see under the microscope. That theory wasn't very well accepted at first, but eventually, of course, as we know now, that it turned out to be true that a lot of diseases are caused by these bacteria and things you can see under the microscope. Well, Pasteur became very famous when he invented the first human vaccine by design. Of course, about 100 years earlier, Edward Jenner had invented the smallpox vaccine, but he really didn't know what he was doing. But Pasteur built upon the ideas of Jenner, and he figured out that what Jenner had done was to take whatever it was that was causing the disease and somehow inactivate it. And when you inactivated it, then you could use that to inject it in a person and they would build up immunity against that weakened disease. And in this picture here, what you see inside that glass jar, that little red thing there, that is actually the spinal cord of a rabbit that had been infected with rabies. And what Pasteur did was he let those spinal cords dry over a period of about a month. And then he would mush it up and inject it into a dog. And then he would take another spinal cord that had only been dried for about 25 days, and then 20 days, and then 16 days, and so on, until finally he was injecting the spinal cord of a infected rabbit that had not been dried out at all. And then what he found, of course, was that this process of injecting more and more virulent rabies virus gave the dog immunity against the virus. Now, of course, viruses hadn't been discovered at that time. They were too small to be seen under a microscope, but Pasteur was smart enough to recognize that even though he can't see it under a microscope, that there was something there. Well, the real success came when the parents of a child, Joseph Meister, who had been bitten by a rabid dog, came and asked Pasteur to use his process on their child. And Pasteur was very reluctant, knowing that something that works on a dog might not necessarily work on a human. But it did, and this was a fantastic success and began really the whole idea behind vaccines for many, many years, that you just take the thing that's causing the disease and inactivate it, and then that can be used as a vaccine. So there are several COVID-19 vaccines that are using this idea. These are, 2 of them are in China, one of them is from India. The idea is that you grow the virus up in some cells, then you purify the virus, and then you inactivate them by a chemical means. It's the same kind of technology that's used for influenza vaccine. And the efficacy of these vaccines in preventing disease is around between 50 and 79%. The next technology to come along was invented by an English scientist, Alexander Glennie. He was trying to find a vaccine against diphtheria. And one of the things about injecting the whole part of the bacteria was that it could cause a lot of side effects. You had a lot of swelling, a lot of fever, and so on. And so the idea was, well, you don't need to inject the whole thing. If you just inject a part of it that's causing the virulence, then you have less side effects and you still get protection. And so they were trying to do this with diphtheria. So in fact, it's the toxin that diphtheria secretes that causes most of the disease. Now, of course, you couldn't inject toxin into somebody. It would kill them, but you can detoxify it by adding formaldehyde. And so this toxoid, when they injected it, they found actually it didn't raise up any antibodies. It didn't work. Well, what Glennie found was quite by accident that he was using a precipitation with aluminum hydroxide to purify the toxoid. And instead of separating the toxoid from the aluminum hydroxide, he just injected the whole thing altogether. And he found that that actually increased the immune response to the toxoid. And in fact, since that time, almost all vaccines have added aluminum hydroxide as what's called an adjuvant to increase the immune response against the subunit. Now, still, even when you do have subunits, there's still a bit of a problem with them, which I'll explain. So here on the left-hand side is a picture of the SARS virus, and you see you have, it's sort of like a ball, and then you have these spikes sticking out of it. And it turns out that all you need to have is an immune response against the spike, and then that is going to give you a protective immune response. But all these other proteins that are there, the M protein, the S protein, they don't help protect you. So you want to just have the spike protein, and that's here on the right-hand side. But again, if you just had this spike protein by itself, and even if you added some adjuvant, you wouldn't get a very good immune response. And so a real trick came about in the late 1980s, and that was to make these things join up into what is known as a virus-like particle. And the hepatitis B vaccine was the first subunit vaccine that used this approach. And so if you can get those subunits to assemble into something that looks like a virus. So here on the right, we see the spike proteins, and they've been assembled into looking like a virus. On the left, here's the actual virus, and you'll notice they're both about the same size and they're both circular. And your immune system recognizes this as, oh, this must be a virus. And so it really gives you a strong immune response. So these two things together then, the adjuvant plus the virus-like particle, both combine to give this type of vaccine, this subunit vaccine technology. And there are two companies working on this. One of them is Novavax, and they've announced their vaccine efficacy was 89.3%. And Medicago is a company in Quebec City
with 76% efficacy.
And they have to add very strong adjuvants. For Novavax, it's Matrix M, and for Medicago, it's AS03, which is an adjuvant system created by GSK. Now we're starting to get into the more modern age, and I want to tell you about Oswald Avery, who is my personal hero. He was born in Nova Scotia, moved to the States, worked at the Rockefeller Institute for his entire career. He made 4 major discoveries throughout his career, each one of which should have earned him the Nobel Prize. Oddly enough, Avery never did receive the Nobel Prize. So in 1923, he discovered that bacteria are surrounded by sugars. And in 1929, he invented the conjugate vaccine. And that is, you simply couple those sugars to a protein, and that increases the immune response against them. And in fact, my entire career was revolved around these conjugate vaccines.
This type of vaccine technology has been used for a COVID vaccine by the Finlay Institute in Cuba, but we have not included it here because we found some problems in the publication which the authors have not responded to.
But he had other discoveries. One of them was the C-reactive protein. And in 1944, he discovered nucleic acids. So, and I just want to mention, by the way, that he was 67 years old when he made that discovery. And Pasteur was 63 years old when he made the rabies vaccine. So how did he discover nucleic acids? What were they, and why was his discovery important? So in the first part of the 20th century, there had been a lot of studies done on genetics. It was learned that the offspring got characteristics that they inherited from their parents. And these things were associated with something you could see under the microscope, the chromosomes. And with doing lots of experiments by combining parents and looking at the characteristics of the offspring, they could even map these characteristics to certain locations on those chromosomes, but they had no idea what these chromosomes were made out of and how the chromosomes contained that information for the characteristics of the offsprings. This is what Oswald Avery's discovery was. He discovered in 1944 that the information that was transmitted was held in the molecules of nucleic acids. Now, the interesting thing about this is that some of you listeners here were alive in 1944. And so all of the things that we know about DNA and nucleic acids and genetic engineering, all of this has occurred during your lifetime and during my lifetime also. So it's really remarkable how we've come along in just 80 years or so. So here's just a brief description of what we know about DNA and RNA. So on the top here, we have the very famous double helix of DNA. And for comparison, I'm going to sort of talk about computers. Most of you sort of have an understanding of computers, and that you have the hard drive, and that was where you store all your photographs and your computer programs, and that's the long-term memory. But then you have also the working memory, and that you have your RAM on your computer, and the more RAM you've got, the more your computer can do it at one time. And so in comparison, that's what the messenger RNA is. It's kind of like the working memory. So what happens is the DNA is copied into the messenger RNA in a process called transcription. And this messenger RNA is the working copy. And then it gets translated by these little machines that are called ribosomes, and it gets the information that's stored in that messenger RNA gets translated into a protein. So proteins are made out of amino acids, and the sequence of those amino acids is determined by the information that's coded in the messenger RNA. So all this is stuff that's been discovered over the last 80 years or so, but even to this day, the way that ribosomes actually do that is still not completely understood, how these ribosomes assemble the protein based on the information that's in the messenger RNA. So there's still more to learn about here, but we know enough that we can actually start manipulating these things, and we have been since the 1980s or so.